All right, let's talk about bacteria. For the next few minutes, we're diving deep into their secret world. And it's a wild one. We're going to uncover the incredibly clever ways they swap and shuffle their genes, evolving at speeds that are just mind-boggling. This is how we get superbugs, the very things that are challenging modern medicine as we know it. Okay, so let's just jump right in. You hear the term superbugs all the time, right? These bacteria that can just shrug off our best antibiotics. But how? I mean, how do they actually get these superpowers? They're evolving at this absolutely blistering pace, way, way faster than we are. So what's their secret? What's the trick? So let's put this in perspective. For complex life, like us, our whole deal for creating genetic diversity is sexual reproduction. We have these complicated processes like meiosis and crossover that shuffle the genetic deck. It's how we evolve. But bacteria, they don't do any of that. No meiosis, no crossover. So how on earth are they mixing and matching their genes to evolve so dang fast? It's a total black box, a real puzzle. Okay, to crack this puzzle, we've got to start with the basics. We need to look at the tools a bacterium has in its genetic toolkit. So let's pop the hood and check out their blueprint. A bacterium's genetic library is incredibly streamlined. It's really just three main parts. You've got the main genome, this one big circular chromosome that has all the core instructions for, staying alive. Then there are these things called plasmids. They're tiny, extra circles of DNA that carry special abilities. Think of them like optional software plugins. And then there's a very special plasmid called the F factor. That stands for fertility factor, and trust me, it is the key to one of their coolest tricks for sharing genes. All right, so we know what's in the toolkit. Now, how do they actually use it to grab new genes? Well, let's get into their first secret weapon. And this one is basically a genetic scavenger hunt. It's a process called transformation. So what exactly is transformation? The best analogy is this. Imagine you're walking down the street and you find a ripped-out page from an amazing cookbook. You just pick it up, take it home, and tape it into your own recipe book. That is exactly what bacteria are doing. They can just slurp up free-floating bits of DNA from the environment, usually from other bacteria that have died and burst open, and then they stitch that new genetic code right into their own genome. It's wild. Okay, here's how it goes down in 3 simple steps. First, some bacterium bites the dust and its DNA just spills out everywhere. Step 2, a nearby bacterium that's in the right state, scientists call this competent, grabs a piece of that floating DNA. And finally, step 3, it incorporates that new DNA into its own chromosome. And boom, just like that, it might have a brand new superpower. So picking up DNA scraps from the environment is pretty clever. But what if bacteria could be, a little more direct about it? What if they could share their genetic secrets on purpose? Well, that brings us to their second method, and it's called conjugation. You can think of it as building a bacterial bridge. Conjugation is a straight-up, direct transfer of DNA from one cell to another. One bacterium literally extends this little tube, it's called a pilus, to physically connect to a neighbor, and then it just shoots a copy of its genetic material right across. It is the perfect bacterial equivalent of airdropping a file from your phone to your friends. It's that direct. And remember that F factor we mentioned earlier? This is where it becomes the star of the show. A donor cell that has the F factor is the one that builds the pilus. It connects to a recipient and sends a copy of that F factor right over. Now here's the brilliant part. The recipient doesn't just get some new genes. It gets the F factor plasmid itself, which means it now has the power to become a donor too. The ability to share genes is contagious. It spreads. And get this whole process is shockingly fast. Scientists actually figured this out by letting bacteria conjugate and then just interrupting them at different times to see what got sent over. In one classic experiment, one gene makes it across in 7 minutes, another one at 23 minutes. And look at this. The gene for antibiotic resistance, it's transferred in just 31 minutes. Think about that. In less time than it takes to watch a sitcom, a bacterium can give its neighbor the secret to surviving our most powerful drugs. Okay, so we've got scavenging for DNA, and we've got direct file sharing. But the third method, this one might be the wildest of them all. It actually involves one of bacteria's mortal enemies, a virus, acting as a clueless delivery driver. This is called transduction. Yeah, you heard that right. Transduction uses a virus as a go-between. specifically a type of virus called a bacteriophage, which is a specialist that only infects bacteria. The best way to picture it is a delivery drone. It's supposed to be delivering a bomb, a malicious package. But somewhere along the line, there's a mix-up at the factory, and it accidentally picks up a box of life-saving tools instead. The virus is just the taxi, delivering bacterial genes from one host to the next without even knowing it. So how does a screw-up like this even happen? Well, a virus invades a bacterium and turns it into a virus-making factory. But during the chaos of assembly, as new viruses are being put together, a piece of the bacterium's own DNA can get accidentally stuffed into a new virus shell instead of the viral DNA. Then this defective virus goes off and infects a new bacterium. But instead of injecting viral code to cause an infection, it injects the genes from the last bacterium it was in. It's a total accident, a biological blunder, but man, is it an effective way to shuffle genes. Okay, let's do a super quick recap. You've got transformation, that's scavenging DNA from the environment. No contact needed. Then there's conjugation, the direct connection, the bacterial bridge, requires contact. And finally, transduction, using a virus as a delivery service. No direct bacterial contact. 3 totally different strategies, but they all have the same result: super fast genetic change. So, okay, transformation, conjugation, transduction. Why should we care about all this microscopic gene swapping? Because what we've just described, this, is the engine. This is exactly what's powering the rise of the superbug. These 3 mechanisms, they're the reason a trait like antibiotic resistance can spread through a bacterial population like wildfire. All it takes is for one single bacterium to get a resistance gene. And then, through these methods, it can just pass that game-changing advantage on to all its neighbors. This is how they adapt so quickly to hostile environments, like, say, a person taking antibiotics. It drives their evolution at a speed that is just staggering. So if there's one thing to take away from all this, it's that these processes, transformation, conjugation, and transduction, they aren't just some weird facts for a biology textbook. They are quite literally the engine of rapid bacterial evolution. And that leaves us with a really tough question, doesn't it? We pour billions into developing new drugs. And bacteria, well, they use these ancient, incredibly efficient methods to figure out a way around them. So in this constant, high-stakes genetic arms race, when you see just how good they are at adapting, who do you really think is winning?